So welcome everybody. My name is Marley Loomis. I was an AmeriCorps member for 2 years at the Port Townsend Marine Science Center and then was a part-time employee and now am a volunteer. Just a couple of housekeeping things before we actually dive into things for the presentation. We are asking that everybody stays muted with your video off throughout the presentation. If you have any questions, which I very much encourage, just go ahead and type them into the chat to everyone, and you'll see that most likely at the bottom of your Zoom screen. And then at the end of the presentation, I'll talk for about 30 minutes first, and then at the end, we will move through some of those questions and hopefully get to as many of them as possible. So please, if anything comes up that you want to know more about or you're curious about, just go ahead and type them into the chat box, and then we will get to them at the end of the presentation. So thank you all so much for joining our special live event for Giving Tuesday today. We are very excited to be participating in this global celebration of philanthropy and are even more excited to get to share with you the story of Tiny, our resident giant Pacific octopus. We are sharing this story today on Giving Tuesday to showcase how donations that are received today can really make a difference. The story of Tiny is an example of the stories that we are telling every day in the aquarium, museum, education programs, outreach every single day to inspire conservation of the Salish Sea. We believe that the more people know about the world below the waves, the more people care about it and consequently want to take action to protect it. We invite you to support Tiny and the aquarium by making a donation today. The link to donate can be found in your Zoom chat. It should be at the bottom of your screen in that little chat window. So please, if you're feeling generous and inspired by this story today, we do invite you to support us by giving a donation. With all of that being said, I'm going to go ahead and take off talking about Tiny, our focal animal of the day. Okay, so a giant named Tiny. Giant Pacific octopuses are the largest octopus in the world, as far as we know. And I'm so excited to get to tell you more about Tiny, our resident GPO. But before I actually give you more details about her and what we know about her, I want to first start by telling you a little bit about how we actually acquired this very exciting animal. We collected Tiny and lots of other very cool animals through a light trap project. So we were working with the Pacific Northwest Crab Research Group, which is a diverse group of crab researchers and managers from the Pacific Northwest. We joined this group in 2020, earlier this year. We started in about March or April working with this group. It's a very broad group of organizations, as you can see by all of the different logos listed here. And this group monitors larval recruitment of Dungeness crab with light traps, which I'll tell you more about in just a moment, with the goal of finding ways to create a more sustainable Dungeness crab fishery. This is the light trap that we work with every day. This light trap, as you can see, is a little bit of a mishmash of different items you might find around the house, in our aquarium, kind of everything all mashed together to contribute to this one common goal. So when we joined this project in 2020, we were trying to monitor Dungeness crab populations using this light trap. And the way that we did that was by attracting the larvae of the Dungeness crab to a light that's inside this light trap. So you can see that kind of tall cylinder that's in the center of the 5-gallon water bottle. And that is the light that attracts zooplankton overnight. This entire contraption is lowered down on a pulley underneath the pier. And then it tracks down that line that's on the right-hand side of the photo. And this entire contraption sits in the water, just resting underneath the surface with the float bobbing on top, and attracts zooplankton throughout the night. The zooplankton then kind of swim in through the funnels that are on the sides of the 5-gallon water bottle. And then as we pull it up in the morning, they are filtered down into the bottom, which is that removable cod end at the bottom. Then in the morning, we come out, collect this big giant water bottle, unscrew the removable cod end off the bottom, dump it all out into a big bucket, and sort through it all. We are looking specifically for the Dungeness crab megalopy, which is a certain stage of larvae of Dungeness crab. But of course, we find all sorts of cool things. It attracts just about any type of zooplankton that's in the area, which makes it very interesting to look at what's inside. Here you can see a graph of the megalopy that we found over the course of this project. It is pretty much in line with what we were expecting from the crab research group. So we had 2 huge spikes in numbers of megalopy. And one of those happened at the end of May, and then the next at the end of June, with sometimes upwards of almost 6,000 megalopy in a day. So that's a bunch of teeny tiny crabs about this big in a bucket that we counted out and then reported our findings to the crab research group. But like I said earlier, we found a lot more than just crabs. So we found so many kinds of different zooplankton that this sample of 9 pictures is a tiny portion of what we found. But basically in terms of zooplankton, there's 2 different groups. You have meroplankton, which spend part of their life as plankton. And then we have holoplankton, which spend their whole life as plankton. Plankton just means that it's something that drifts with the currents and cannot swim against the water. So a jellyfish is an example of holoplankton because it spends its entire life drifting with the currents. An example of meroplankton is Dungeness crab larvae. So crab larvae floats along with the currents, then it settles out onto the bottom. It's no longer plankton, but it started out that way. So we found some really surprising species in this project. If you can see this one, the center left and the center picture, those 2 that kind of are clear looking. I don't know if you can see my mouse or not, but they're kind of clear looking. Those 2 are actually a type of amphipod that both live in really deep water, and we found them at about 6 feet of depth. So we found some very interesting things. In order left to right, top to bottom of these images, we have a tadpole sculpin, which we did indeed raise in the aquarium, a jellyfish, some barnacle nauplii, a couple of crazy worms, those 2 deep water amphipods. On the bottom left, those are 2 different types of crab megalopy. Neither of them are Dungeness, but they're different crab megalopy. A little larval fish and a copepod. So we were finding a huge diversity. One of the interesting things about this project is that the assemblage of larvae and plankton that we were finding was very different from the makeup of juveniles and adults in terms of species that we see around the pier. So that was interesting in itself. And then we were able to use this project for tons of different types of outreach and education projects. We had school groups come in or Zoom call in for a remote class to see these different creatures. We were posting them on social media. So we were trying to come up with different ways to use these very interesting animals as a part of outreach and education to fulfill our mission. A few of these species were raised successfully in our aquarium after we collected them, Tiny, of course, being one of them, and I'll get more to her in just a moment. We also raised a bunch of buffalo sculpin, a bunch of grunt sculpin. If you've been to our aquarium and have seen those fun little fish, we raised 13 of those from the light trap. So we were able to contribute to our aquarium collection with this light trap. And then finally, we saw some very interesting seasonal variation with the animals that we found in the aquarium. So this was an optional part of our light trap study. It was not required by the crab research group, but we found it very interesting to record the presence and absence of some different species that were frequent visitors to our light trap. On the left-hand side of this table, you can see the names of some common fish and octopus that we found in our light trap. And then as you move from left to right, that's when we saw the presence of these different animals. So as you can see, a lot of the fish, most of the fish that we were seeing were in April and May. And then the surf smelt really picked up towards June, July time. But we really ended up seeing octopuses in our trap almost throughout the entire project. We saw them on the first day, and then throughout the whole summer, we were seeing these octopus paralarvae. So the animal that you all have come to learn about today, our octopuses. Like I said, we saw these through the entire project, which was always fun, always my favorite thing to find in the light trap. In this area, we have two main species of octopuses, red Pacific octopuses, which are generally a lot smaller, and giant Pacific octopuses, or GPOs, which is what most of the time what you're seeing in larger aquariums. We found mostly red Pacific octopuses, but most of them were pretty small. So in all of these 4 pictures here, you can see a lot smaller octopuses than Tiny was when she came in. The photo with my hand underneath the dish, those are all red Pacific octopuses, and it gives you a bit of scale as far as how tiny these are. There were something like 9 individuals in that small dish. And then the little spots are their chromatophores, or what makes them colorful. Octopus paralarvae are notoriously hard to raise. They are pelagic, meaning they are swimming in the open water, which is why we were able to find them in our light trap. They do not settle out onto the ocean floor like you might imagine until months down the road. Giant Pacific octopuses settle after 5 or 6 months of age. So that whole first half a year, they're just floating around in the water, very small. Because they are floating in the water and they are so fragile, the paralarvae and the juvenile, or the really small juveniles, require something called a chrysal tank, which is something you may have seen with jellyfish in it. It's a circular tank with circular flow, no sharp corners, and it's a safer way of keeping these little guys circulating. We do not have a chrysal tank at PTMSC. We do not have a jellyfish tank, so we were not able to collect these pelagic paralarvae because we don't have the appropriate setup for them. So all of these guys were released back into the water after finding them in the light trap. Tiny, however, was different. So in this video, you can see Tiny. She is the biggest one in this video, right next to a little piece of krill. That's what that little floating object is. And then earlier in this video, and in a little bit, you'll see there's 2 other octopuses in there with her. All of these 3 were giant Pacific octopuses. And you could see Tiny is kind of settled onto the bottom. So at this point, when we found her, she was already benthic, meaning we're estimating she was around 5 or 6 months of age. She had settled out to the bottom. So this meant that for us, we did not need a chrysal or a jellyfish tank. Most likely we could keep her in one of our existing tanks. She crawls along the bottom, and this was on June 10th. So this video is from the day that we found her we nicknamed her Tiny, and then we found the two other ones that were even smaller. So we called them Tiny, Tinier, and Tiniest. Tinier and Tiniest were released later that day because they were not as settled and as mature as Tiny was. Okay, so we have Tiny now at this point. After June 10th, we're like, okay, I think we can manage it. We might be able to raise this little octopus. How are we going to do it? What are we going to put her in? What is she going to eat? How's this going to work? So we had this existing setup, basically, from raising larval fish. So this is our larval fish tub. It is a very fancy 40-gallon plastic muck tub with a hole drilled in the side, acting as the drain. And then this drain cover is a pipe with some holes drilled in it, covered and glued with a mesh screen around it. And then the brownish hose that you see coming in from the top is the water supply. So this muck tub seemed like a sufficient tiny tank because she was settled on the bottom, so we didn't need the circular flow. We lowered our water level way down to keep her from wanting to climb out, and kept a variety of different decorations in there. So you can see there's a plastic octopus tentacle in there, which is hollow. So we figured she might want to crawl in there at some point. We had some grass, some barnacles, rocks, things like that, just to give her sufficient hiding places. Kept it clean, kept it stocked with food. A lot of the food at this point, most of the food at this point, was coming from the light trap. So that same light trap that we got Tiny from was also providing her food source, which was mostly zoe, which is the first stage of crab larvae, crab zoe, and other small crustaceans. And we would just put more in there every day, hope she's eating it. Saw some of them disappearing, but she was very much still nocturnal at that point. Stayed hidden. So this was a hands-off phase. We did not want to stress her out, wanted to just make sure she was eating, surviving without too much disruption. This next phase, monitoring, we started once we realized, okay, she's eating sufficiently, she's big enough to be weighed, and we need to be keeping closer tabs to make sure she is sufficiently active, growing, and developing. The process here is to pull her out of her tank weekly to monitor for growth and development. The way to do that is usually she is hanging out in her favorite barnacle or oyster shell or whatever her favorite home is at the time. So the easiest way is to just pull that whole home out of the water, then coax her into this small tiny weigh cup. And she kind of slips out of her barnacle and into the tiny weigh cup. And then we dump the water out and weigh Tiny while she's damp. Octopuses can still breathe as long as they are still wet. So we did this step very quickly. We weigh her on this scale, add the water back in so that she's not out of the water anymore, and then subtract the weight of the cup. So that's our very scientific way of monitoring for her growth. We were also able to observe significant changes, both behavioral and physiological. The interesting thing here is that when we first collected her, she looked like a tiny, clear ball of jelly, essentially. And then as she developed, she became more opaque, more colored, started developing textures, was showing different behaviors, things like that. So this is the time when we really are able to document how she's developing, which is very fun to see her develop the classic GPO behaviors and development that you would expect. And then at this phase, we also started introducing frozen foods, which mostly was krill and shrimp. This is an essential step because If she's only eating live foods, sometimes they're harder to secure. It's harder to secure live foods. We need to make sure she has a varied diet, just like everything in our aquarium and all of your pets. And, you know, you want to make sure they have a varied diet with enough minerals and vitamins in their foods. And it also was essential for her enrichment to know that we could engage with her to give her that frozen food. And then phase 3, which is our current phase, is the exhibit phase. So octopuses, as I'm sure you have all heard, are really great escape artists. They can sneak through just about any hole as long as their beak can fit through it. But if you can imagine, Tiny is very tiny and has a very tiny beak. So any little space is a possible escape route. AstroTurf, or fake grass, AstroTurf, is a really common material to deter wandering octopuses. So as you can see on this top photo here, this is Tiny's tank, which has that AstroTurf lined along the top and also wrapped around the water inflow, which is on the right-hand side. They don't like to stick to it. I don't know if they don't like to stick to the AstroTurf or if they just can't stick to it with their suction cups, but either way, they don't sneak through it. So we have wrapped the top of her tank in AstroTurf, and then we have a weighted lid with all holes sealed off to keep her nice and contained. We don't want to come in the morning and find any wandering octopuses on the floor. We also vary her tank furniture every week. So when we pull her out to weigh her, we make sure her tank is nice and clean, switch up the furniture that's in there, give her a different den, or usually we keep her favorite den in there, which most recently has been the barnacle that she's on top of in the bottom left corner. But we also add in some artificial kelp pieces, occasionally some little toys, different dens, things like that. And then we also need to continue trying to entice her by frozen foods. So right now, her favorite food is purple shore crab, which we collect on the beach. Right now, she's getting shore crabs about an inch across from leg to leg. And she's still not very excited by frozen food, although recently she did start taking more shrimp again, which is great. We continue to monitor her weight. We weigh her weekly still, which is a very important part of her enrichment as well. So enrichment is something that a lot of zoos and aquariums do to keep their animals happy, healthy, stimulated, to make sure they're not getting bored. For octopuses, a lot of times that includes puzzle feeders, toys, things like that. For Tiny, we are just now starting to introduce different toys, but the weighing is also a form of enrichment. It's changing up her environment, and we weigh her weekly so that she can get more used to it. If we just weigh her once a month, it's more of a disruption to her daily behavior. If we weigh her weekly, we can keep better tabs on her growth and development, but we also are able to kind of keep her accustomed to the process so that it's less jarring. Okay, so Tiny now, she's in her exhibit, she's going smoothly, everything's good. How do we know that she's a GPO? When they are paralarvae at this stage, juvenile octopuses or paralarvae are distinguished by their chromatophore patterns. So the dots that you see there are called chromatophores. It's their color changing cells. You can actually see them contracting and expanding if you look at them when they're really close up or when they're really small. The easiest way to tell the difference here is that a GPO has one row of chromatophores on their tentacles, so one row of dots going down their arms, and red Pacifics have two rows of dots going down their arms. So I'll give you a second, see if you can figure out which picture is which. One is a red Pacific and one is a GPO. So if you can see the right-hand photo on your screen, there's 2 dots at the top of each arm and 2 rows going down each arm. So that one is a red Pacific on the right-hand side. The left-hand side is 1 row going down the arms. That's also an older individual. That is actually Tiny. That picture right there is our resident GPO. So you can tell this from a very young age. That image in the center is showing that you can also distinguish them by the chromatophore patterns on their mantle, but it's a lot more complicated. We can see from their arms which one is which. We also can tell her sex from a relatively young age, which is very cool. The trickiest part about sexing an octopus is when they're very small, their suction cups are also very, very small. So male octopuses have a hectocotylus on the 3rd right tentacle. So that schematic on the left-hand side is showing you which is the 3rd right. And then the blue circle on the picture of Tiny is showing you her 3rd right arm. So on that arm, if they have suction cups all the way to the tip of their arm, then they are a female. If they do not have suction cups all the way to the tip of their arm, then they are male. And that hectocotylus is, looks kind of like a spoon almost. A little spoon-shaped groove is in the center of it. And that is how the male delivers the sperm packet to the female. So on males, they do not have suction cups at the tip of their arm. Tiny does, so she does not have a hectocotylus, therefore she is female. Okay, so this graph is showing you the blue line is Tiny's weight by week, and then the red line is showing you the percent gain daily. The biggest thing is that we want to see an overall trend of increased growth. Octopuses can retain water in their body systems, so when we weigh her, there is a chance she's retaining more water that week. Maybe that's why she's gaining weight. So overall, we want to see an increase, but there is some fluctuation from week to week. Her average daily growth rate is just under 2% at 1.97%. Similar species are at about 2%. So we're right on track there, and she's still consistently growing. It's very new, that all of this territory is very new. There are only 2 documented studies of raising GPOs successfully in captivity. So a lot of the research that we are doing to successfully raise Tiny is based a lot on other species or on later growth of GPOs. So a lot of this is still pretty uncharted. All right, so this is a little bit of a video about Tiny's growth. So you can see at the beginning, she's very much a clear little blob. That's the same cup that we weigh her in weekly. So when she grows to fill more of that cup, you can really see the difference. And that is intentional. It's a better way to see her growing. Then at this point, you can see she's really becoming more opaque, showing a lot more chromatophores than you would see at the very beginning. This is the first week that we really saw her developing darker colors. And you can start to see the development of her web between her arms. And then you can see here in that picture, she's developing texture. So now she's really turning darker colors, developing that kind of bumpy exterior that you come to see in the adult octopuses as they grow. And then of course, her mantle starts taking more of a prominent part of her body shape. Some really different colors coming into play at this point. And then there's that texture that's really outlined in that picture nicely. This is when we release her back into her tank at the end of weighing. we like to let her go smoothly back into her tank. So this is the release post-weigh. At this point, she's really filling up that same cup. And we're able to see her out and about a bit more in her tank, which is really fantastic to see. This is one of the forms of enrichment. We have a little floating log for her. And that's our most recent weigh-in photo. And there she is going back into her favorite little hiding spot. So we thank you all so much for supporting us. This is our most recent weigh-in photo of Tiny. And you can really see she has some crazy interesting coloration happening here. We will be upgrading her weigh-in cup very soon to make it more enjoyable for her. But we kept her in that same smaller cup just for a short bit so that we can see the development. But thank you all so much for coming. And I would love to take some questions at this point. I saw a few of them pop up. And I think Liesl is going to hopefully help me out in moderating.
Yes, I am here, so I can help with the questions. Some of them were already answered, Marley, in the end of your presentation. Great questions. One that wasn't answered was John Petrata asked, do we thaw her food out before feeding her if it's frozen?
Yeah, that's a really great question. We have tried both ways. Eleonora, our last GPO, really enjoyed frozen foods and did not like thawed food very much. So we have tried both ways to see if Tiny has a preference. So far, she does not really like frozen food, and so we've been thawing it first. But I'm interested to see how that develops since Eleonora really loved her frozen food a lot more.
Mm-hmm. Yeah, and so there's another question here about companionship. This is a real common question, whether GPOs like to have a buddy. This is from Linda Herzog, if you could address that.
Yeah, it's a common question. GPOs and octopuses, almost all species, are very solitary animals. So they like to have their own den. They like to have their own space. We don't really keep any octopuses together in captivity. It can lead to increased aggression between the two individuals. So they definitely are solitary. When it comes time for reproduction, we will, assuming we are continually successful with Tiny, just as we did with Eleonora, she will be released to hopefully find a mate. But they definitely are kept solitary to keep aggression down in their tanks.
Great, we have some really good questions coming in here now. One of them is, do you plan to get a specialized tank to be able to have more GPOs as babies in the future?
It depends a little bit on how this goes with Tiny, for one. At the moment, we do have, if you include the fancy muck tub at the beginning that I showed you, we have now 5 octopus proven tanks, all of different sizes. So we don't have anything that was built with the intention of keeping an octopus, but we have sort of retrofitted them to be able to hold octopuses safely and in healthy ways. So if we were to raise more in the future, we would hopefully be prepared to do so.
Since you mentioned Eleonora, the GPO we had last summer, there's been a couple questions about her, whether we have any information on how she fared after her release. And then a really great question, is there any possibility that Tiny could be related to Eleonora?
Yeah, so as far as what we know about Eleonora, we're not really sure. There's no real way of going back and monitoring really what happened. We did go back and check a short while after she was released, and she was no longer in that den after a little while. So it's possible that she moved on to another place. If octopuses are not quite ready to lay their eggs yet, it's common that they kind of move around and try out different dens, find different octopuses, things like that. So it's possible she just moved to a different spot. It is possible, I guess, that it could be Eleonora's little offspring. I'd like to think that. I think that's pretty fun, but honestly, there's no real way to know. The females die after their eggs hatch, so there wouldn't be much of an overlap to see if it was Eleonora's offspring, but I don't see why it couldn't be possible.
Seems like a nice thing to think. It is very possible. We have also a couple questions about how long do GPOs live in captivity in the wild? Does that differ at all?
It doesn't really differ. They live 3 to 5 years, which is a longer lived species of octopus than most. GPOs have a longer lifespan. So that lifespan is based very much on their instinctual behavior. So they will mate, the females will then lay their eggs and then care for their eggs until they hatch. The entire time that they're caring for their eggs, they don't eat. So even in captivity, if they're offered food, even if it's offered right in front of them, they will not eat. So it's sort of an instinctual timeline, 3 to 5 years. Tiny, it's hard to know how old she is. We're thinking she's somewhere between 9 months to a year, just based on when we know that they settle out to being benthic. But yeah, it's a pretty ingrained time frame.
And then also regarding her personality, do you see it starting to develop at this 9-month stage, or what have you noticed about her personality, especially compared to Eleonora?
Yeah, one of the most fun parts for me has been watching Tiny develop kind of classic GPO behaviors. So I've also worked with some red Pacific octopuses, if you remember Paprika and Rhodey that we had last year. And they move in very different ways. So GPOs kind of move with their arms first and kind of crawl along the bottom. And the red Pacifics kind of tuck in all of their arms and move like a little dumpling along the bottom when they're kind of caught out in the open. And so it's been really fun to watch Tiny develop that same crawling behavior that we've seen in Eleonora. I do think that she is a little bit sassy with us sometimes. She'll blast some food out of her den if she doesn't want it with her jet and things like that. So it's really fun to watch it develop, but it is definitely in the beginning stages of seeing her personality, since we are just now kind of introducing more interactive forms of enrichment instead of just ensuring her hunting and survival.
Okay, 2 more questions here. What are the natural enemies of GPOs? And is it possible to take a DNA sample for later analysis?
Yeah, as far as, I'll answer the predators question first. So GPOs have lots of predators. Octopuses in general have lots of predators. They are one big squishy muscle with a beak in the middle. So anything that can kind of grab hold of them is definitely a predator. GPOs in particular, lingcod really like to eat octopuses, seals, sea lions, things like that. Anything really, as long as they can catch hold. The juveniles are very they hide a lot more. So if you can imagine, if you were a teeny tiny little muscle mass, you'd probably want to hide out a lot more. And they do have a venom that they can inject with their beak, but yeah, they have many, many predators. As far as taking a DNA sample, that would be very interesting. I don't know. I don't see why it wouldn't be possible. At PTMSC, we don't really have the resources or justification behind doing that. But I don't doubt that it could be done in a research lab somewhere. That'd be very interesting to see, just to learn more about their lineage.
Okay, it looks like there's just one more question, which is about if Tiny's care is regulated in any way by state or federal government. And also just if you could speak to the general way that octopuses may be protected or not protected in a formal way.
Yeah, octopuses are, because they are invertebrates, they don't hold a lot of protections. Vertebrates in general, in terms of life sciences, carry a lot more regulations as far as being allowed to keep them in labs, things like that. Octopuses are, because of their intelligence, they are kind of on their way to collecting more protections, but they still are kind of in that invertebrate section, so they don't have that many protections on them. As far as regulating her care, like I said, it's pretty, we're in kind of uncharted territory as far as raising a juvenile. So we are basing our plan of care very much on other species and just what we know in general about GPOs. So a lot of that is just ensuring proper enrichment, lots of environmental changes, variety in foods, things like that, not overstressing them, and things that we know to be true about keeping all octopuses in captivity. So it is very interesting to record what we're doing because it is a newer field.
Okay, there a couple more questions did come in. We're at 43 minutes, so I think there's time to take a few more. So we'll see how many more we can fit in. First one is this going to be recorded? And it is being recorded, so we will be posting it on YouTube for people to see later if they weren't able to be here live. There's also a question, are GPOs prolific here in the Salish Sea?
Yes, we do have a good number of GPOs here. The Puget Sound in general is a pretty common spot to find GPOs. Because they are solitary, you won't really find a whole cluster of them in one spot, but they are definitely, there are plenty of them around as far as we know.
And I'm just going to add in that Allie Redmond, who's participating as an audience member and is our wonderful aquarium curator on staff with the Marine Science Center. Thank you, Allie, so much. She had a lot to do with this presentation as well. Did want to remark that there is a permit required to collect and house a GPO in an aquarium. So we, of course, have that permit in place and do follow all the regulations. And then there was a question just related to the DNA answer that you were giving before about what about freezing, like extrusion, body waste for later analysis? Is that kind of thing possible?
I don't know, to be honest. I'm not sure. I don't see why it wouldn't be possible. At this point, we don't have a good enough reason to do so. Yeah, it would be an interesting project to look into further, but I don't know of anybody doing that specific type of study. But it would be interesting just to learn more about them. Yeah.
Yeah, and we invite everyone in the audience who may have knowledge or awareness of partners that we might work with who would be interested in, participating with us on a research project to let us know. We definitely would love to reach out. So that is the end of the questions. So I think we'll wrap it up and say a huge thank you to Marley and to Allie for creating this presentation. and presenting it so beautifully. And thank all of you for participating. It looks like we had about 46 people on the call here today. And we thank you so much for your interest, for spreading the word about Tiny and the story of her. And we hope the presentation gave you some great ideas about what donations coming in on Giving Tuesday are making possible. And if you're interested in supporting that, I'm going to drop the donation link one more time in the chat, and you're welcome to click on that if you want to make a donation, or of course you can just go to the website and do that there as well. But we are so grateful for all of you for making this possible, and thank you for sharing in the story of Tiny. And have a great Tuesday. Thank you so much for coming. We're going to just go ahead and end the meeting at this point. Thank you again.
Thank you.